How's it going, everybody? It's Jeffrey here, back at it to teach you another physiology topic. And today we're going to be going over skeletal muscle contraction. So basically what I'm going to be teaching you guys today is how physiologically your body allows you to do this. Your concentric contraction and your eccentric contraction. But before I move on to the pathway, I just want to say that this first part of the pathway really focuses on how the neuron passes an action potential to the skeletal muscle. And if you guys have any trouble with how an action potential is propagated along a neuron, you really should check out my previous video, because I really go in depth on how because it's pretty much just about how an action potential is propagated along the neuron. So yeah, you guys should check that out before you go into the skeletal muscle contraction if you're confused at all on how an action potential works. But with that being said, let's get started. So the first part of the skeletal muscle contraction pathway is we're going to start at the neuromuscular junction. And so we have this picture here, right? And we start off with the somatic motor neuron. And at the somatic motor neuron, we have an action potential that's being propagated down towards its axon N-terminal. And once this action potential reaches its axon N-terminal, voltage-gated calcium channels are then opened, allowing calcium ions to flow into the cell. And once these calcium ions flow into the cell, these vesicles filled with a ligand called acetylcholine then are brought down to the membrane, and through a process called exocytosis are then released from the cell into the synaptic cleft. And once these acetylcholine molecules are inside the synaptic cleft, they are then eventually brought down to acetylcholine receptors that is found on the motor end plate of the sarcolemma of the skeletal muscle. And the sarcolemma is the membrane of the skeletal muscle. So once these acetylcholine molecules bind to the acetylcholine receptors, an action potential is then propagated along the sarcolemma and then brought to the next part of this pathway. So now we have this action potential continuing along the sarcolemma and eventually being brought into a hole where a T-tubule is contained, or what we call a transfer tubule. It's the same thing. And as this action potential travels down the T-tubule, the sarcoplasmic reticulum is then stimulated, allowing for the secretion of calcium ions. And those are that orange dots that I have in the picture. And these calcium ions are then brought down to the myofibril that is found inside of the sarcoplasmic reticulum, or under it, which is this line I have here that's shown underneath the sarcoplasmic reticulum. And within the myofibril, that's where our pathway continues. So in part 2 of the muscle contraction pathway, we're going to be looking at myofibril. And within myofibril, we're going to be talking about the sliding filament mechanism. So within the myofibril, we have two proteins. We have myosin and actin, which is these circles here, and then this branch-looking protein here. The myosin is the bigger one, and the actin is the smaller one, which is something you should keep in mind for these two proteins. And these myosin and actin proteins are grouped together in a functional unit called a sarcomere. So what basically myosin and actin does is myosin binds to actin and pulls on it, allowing for these two actin proteins to come closer together. And this action of binding and pulling is what we call a power stroke. And so allowing these two proteins to move, these two actin proteins to move closer together, is what basically contracts your muscle cells. And however, there's more detail to that, especially when we look at actin. So I want to move over to start looking into the actin protein. So within actin is actually made out of two other proteins. We have tropomyosin and we have troponin. Tropomyosin is those strand-like proteins that cover these gray holes that are found on actin, and these holes are called the active sites on actin. And the troponin protein is a protein that's attached to the tropomyosin that plays a big part once calcium is put into the mix. So remember the calcium that was secreted by the sarcoplasmic reticulum? See, now these calcium ions are then bound to these troponin proteins, allowing for the troponin protein to move down along with the tropomyosin protein, now exposing these active sites on actin. And once these active sites on actin are exposed, we then myosin is then now allowed to bind to the protein and do the binding and pulling, or the power stroking. And also, you should also remember that the act of binding and pulling doesn't require any ATP. However, when myosin has to be unbound from or detached from the actin protein, ATP is required to remove that myosin protein. And that's basically how a muscle contraction works. Okay guys, so I just want to go through one more thing about the sliding filament mechanism, and that's the I band, the A band, and the H zone. So what these are is basically when the muscle is relaxed, the I band will only contain actin, whereas the H zone will only contain myosin. However, the A band is special because whether or not the muscle is contracted, the A band will contain myosin no matter what. And this is because the A band will always span the length of the myosin protein. So once this sarcomere is then contracted and these two actin proteins are brought together, the I band will disappear and the H zone will disappear, but the A band will still be there. And yeah, that's pretty much it. So I just wanted to do one last thing with you guys, and since this pathway is pretty short, I just wanted to run through it super quickly and just act as this like a final wrap up kind of thing. And what you can do is you can pause the video here and then test yourself, see if you know the whole pathway by yourself. And once you play this video, you can see how well you did compared to how I explained it. So we're going to start off with the somatic motor neuron, and there's an action potential being propagated down to the axon N terminal, where voltage-gated calcium channels are then opened, allowing calcium to go in, and these calcium ions then allow ligand filled with acetylcholine to be brought down to the membrane, and through a process of exocytosis, the acetylcholine molecules are then brought into the synaptic cleft, where they are picked up by acetylcholine receptors on the motor end plate of the sarcolemma. And once these receptors pick up the acetylcholine, an action potential is then propagated, and once this action potential is propagated, it is brought along the sarcolemma and is eventually dropped into a hole where a T-tubule is. And once the action potential drops down the T-tubule, the sarcoplasmic reticulum is then stimulated, allowing for the secretion of calcium ions. And these calcium ions are then brought into the myofibril, where it is picked up by actin. And actin, through one of the proteins it's made of, troponin, the troponin picks up the calcium ion, bringing down the tropomyosin, exposing the active sites on actin, allowing these myosin heads to bind to the active sites on actin and allow it to bind and pull, which is a power stroke. And in this act of the power stroke, that's how the muscle is contracted, bringing these actin proteins closer together. And keep in mind that when the myosin binds and pulls on the actin, that doesn't require ATP, but the act of the myosin detaching from the actin protein, that's what causes ATP. And that's basically the skeletal muscle contraction pathway. So I hope you guys enjoyed the video. This one was a lot shorter compared to the other pathways, but it was still fun nonetheless. And thank you all for watching, and I'll see you all in the next video. Contraction, mostly that, eccentric motion, just because gotta tear those muscle fibers apart, if you know what I mean. If you know, you know. Oh my, oh god.